I'm thrilled to be introducing Dr. Marie McDonald, who is the Chief of the Diabetes Section and Director of the Diabetes Program at Brigham and Women's Hospital for this very important panel on one of the most pressing of all chronic diseases. Dr. McDonald.
Thank you so much, Chris. It is really a pleasure to be here with all of you today, and I'm going to start by introducing this really illustrious panel as we discuss really the cure for insulin deficiency in 2021. We'll start with Tom Ballenbeck. Tom is the CTO of Advanced Regenerative Manufacturing Institute. He's the Chief Technology Officer, as I mentioned. And prior to ARMI, Tom served as Vice President of Research and Development at Harvard Apparatus for Regenerative Technology. He's really at the forefront of developing and cell manufacturing of biomaterial-based cells and implants. And before that, he was at Organogenesis. So we welcome you, Tom. Next, we have Mansi Jaiman. Mansi is the Vice President of Clinical Development at ViaCyte. Mansi comes to us with the unique background of being a pediatric endocrinologist. She has significant expertise in both drug and device development, especially for diabetes. So she has, prior to ViaCyte, she was Senior Medical Director at Covance Clinical Development Services. She had, prior to that, worked at Harvard Medical School developing clinical tools for type 1 and type 2 diabetes pediatric patients. And she's been on the side of developing work in the bionic pancreas field. So she has that unique look at what we have in technology, on the technology side of diabetes management. Next, Bastiano Sana. Bastiano is, he joined Vertex in 2019. He's currently the company's Executive Vice President, Chief of Cell and Genetic Therapies. He's overseeing Vertex's cell and genetic therapy and the new Vertex Cell and Genetic Therapies research site here in Boston. He was previously the CEO of Semma Therapeutics, which was acquired by Vertex in 2019. And under his leadership, Semma's pioneering work to develop cell therapy treatments for those living with type 1 has advanced significantly, and we are looking forward to hearing from him. And finally, Rogerio Vivaldi. Rogerio is the CEO of Sigilon Therapeutics. He is the MBA and President and Chief Executive Officer of Sigilon, which has been involved in cell engineering to produce efficient or therapeutic proteins in humans. And he's in partnership with one of his products is in partnership with Lilly to develop a cell-based product for type 1 diabetes therapy. Dr. Vivaldi is also a physician for 25 years. He is well known as a patient advocate. So he's on both sides of the story, as both advocating for the patient as well as developing what we think what he hopes to be scalable therapeutics that will be globally accessible. Okay, so welcome everybody. I'm glad you're here. We're going to start. I'm going to start just by opening the topic. Kick us off with a clinical perspective. So we know we're talking about type 1 diabetes. Our discussion is about curing severe insulin deficiency. We know that managing this disease requires currently painful finger sticks to test blood glucose levels, insulin injections, when accessible insulin pump devices that deliver insulin continuously, and even integrated with glucose monitors. But even these devices, as advanced as they are imperfect. And patients, their families, their physicians have sought a better way to manage this disease. There's also the cost of insulin that patients face, estimated to be about $200,000 over the span of a lifespan of somebody with type 1 diabetes. The cost of an education, the cost of a home. This excessive cost over the lifespan is also something to consider when we're thinking about the inefficiency of current therapies. And we know that as an alternative treatment, islet replacement therapy has been developed over the last 2 decades using cadaveric islets, but the limitations there are clear, although we have spent time learning about the limitations of that therapy in terms of the immune system and cloaking against autoimmunity, which is fundamental in type 1 diabetes, for example. But finally, we're here to talk about the next step, which would be differentiating pluripotent stem cells to functional beta cells. And this technology is being developed by you all and others around the world, but really you are at the forefront, and we're hoping to understand from you where we're at, in that therapeutic space. So to start off, I'm going to start with you, Mansi. You know, since you have been at the on the technology side, what have been the most important advances in the past 5 years in diabetes therapy to get us where we are today? But why, in that light, do we still see that a functional cell therapy is the next step for the field?
Sure. Thank you for that. So, you know, as a clinician, from a clinical perspective, treating patients themselves, I've seen improvement in all the devices that are involved in their diabetes care, improvements in blood glucose meters, continuous glucose monitors, even smart pens, specifically over the last 5 years, and most recently, closed loop or hybrid closed loop systems. Now, as a clinician, I want to provide the best therapy for my patients, decrease the burden of care. And as these devices have advanced, so did the improvement in their care. But technology has limitations, and it still requires external devices that need to be attached to a person. It's a reliance on pumps and sensors functioning properly, being able to dose properly. And it's user input. There's still user input day to day. And so I think the technological advances are certainly temporal, and they have changed the space. However, functional cure would really mean that we eliminate the external devices, the user input, and eliminate that patient burden that we're all striving to do.
That's great. I'm just going to open this up to the others. Maybe you have other perspective, maybe Rogerio specifically, but any of you, please.
Yeah, and first of all, thanks. It's the first time I'm attending not virtually to a meeting after more than a year, so it's great. It's a different world. I think that, Mansi, you said very well in terms of lots of advances. Again, I have type 1 diabetes myself for over 47 years. I do remember how it was 47 years ago. I do remember. I was 10 years old. But it's still, the limitations are very important. Closed loop system may control your glucose in about 67, 75 minutes. That time is going to be a challenge for all the chronic complications and all the future for that patient. If we, through our cell therapy, to a really advanced cell therapy, we can shorten this time of correction to something between 15 and 20 minutes, the patient lives will be different. And that is what excites me to be there.
Yeah. So, you know, on that note, let's get into the science a little bit. Bastiano, Dr. Sana, you've been at the forefront at SEMA and now at Vertex, just in differentiating the cells and dealing with the barriers to get to the next step. Could you tell us a little bit more about the critical barriers in terms of differentiation of cells, but also autoimmunity, the matching the cell to the patient, or the biology of the patient, and then touch on engineering and manufacturing?
Absolutely, it would be a pleasure. Maybe I just want to maybe say a few words on a concept that both Rogerio and Manasi have
Please.
Really explained clearly, and maybe in a different angle. This year will be 100 years from the discovery of insulin. And recently I went back and read the Nobel lecture that Dr. Banting gave, accepting that prize. At the end of the lecture, he did say, insulin is not the cure. And that was 100 years ago. And 1 century after, we're still asking patients and their families to manage their disease with insulin. Now, of course, we have much better tools and technology, but we're still asking the patients to manage their disease with insulin. We don't isolate insulin from pig parts no more, but the concept does not really evolve much. So that is when I think we start talking about functional beta cells as a regenerative medicine approach to give back to the patient something that is not insulin, is actually the solution that nature intended, the beta cells. Speaking about the technology, we have been really busy and focused in advancing technology. Of course, was developed over 20 years by Dr. Dr. Melton in his lab, and we believe that we now have a type of beta cells, actually is Islet, because we also do alpha and delta cells, that is virtually indistinguishable from cadaveric islet. Like you said, one of the major limitations in cadaveric islet transplant, which by the way, when it works well, is availability and quality of those cells. So I think that the greatest barrier has been availability and the scale up, the ability of producing those fully functional, terminally differentiated cells at a scale that is available to a large number of patients instead of the very few that cadaveric islets can be used today. So I think the biggest barrier over the next few years, and we are busy doing that, is the scale up. Be sure that we can produce reliably large quantities of those cells that need to be qualified and need to be of the highest quality possible. Our first program is, of course, just trying to address the issue of cells. You mentioned the issue of immunity, which for Vertex, the first trial will be a recapitulation of the islet transplant paradigm. So we're going to be using immunosuppression, but that is, of course, not the end game. We think that immunosuppression might be appropriate for a few patients, but not for the entire field of type 1 diabetes. That's why we have focused of the last years on encapsulation technologies, developing a new concept of encapsulation that would protect the cells from the immune attack, including autoimmunity. And we're going to be testing that in the clinic very soon. So it's an exciting time. We are not the only ones, of course, pursuing this approach. And that is, I think, is a great thing for patients and the family, that the field has moved, not just us, but the field has moved on. And now we are at the verge of really bringing that promise to delivery.
It's very promising. I agree. It's exciting. And I'm glad you brought up encapsulation and the autoimmunity issue. Rogerio, maybe you could take us to the next question about just cell survival before we think about scaling up. What are you thinking? How are you addressing the issue of cell survival in the patient?
And thanks, Bastiano, for reminding us about the 100 years of insulin. Again, can you imagine treating a patient with a completely new approach to deliver functional cure for patients with type 1 diabetes 100 years after? Again, Banting Investor had done that. Let's think about cell survival. One, cell therapy since the 80s, we saw cell as replacing, turning cells into medicine. Problem is that the cells, as an allogeneic cells, they will trigger immune response. So encapsulation could completely clear this problem. The problem is that the encapsulation in the past was triggering a perifibrotic overgrowth, which will create a fibrosis into that capsule that will avoid the cells to be nurtured, and then cells will die anyway. At Siglum, we developed a shielded living therapeutic platform that is based on Bob Langer work with Dan Anderson at MIT for over 10 years, where they did a screen and found families of small molecules that once conjugated with algenates, they could really give this afibrotic component to the sphere. And we don't say capsule, we say spheres. Our spheres have two components. The outer the compartment is where this fibrin is, and we demonstrated that the macrophage, they don't recognize the sphere. They go, and we see that a macrophage as the first step for any immune response. In the inner compartment is where we optimize over the past 3 years to make sure that we have the best environments for cells to survive and survive happy, so that they can be producing. Again, the sphere has a porosity that allows by diffusion the nutrients, oxygen, the nutrients to come in, and then the products could go out. In case of diabetes, will be insulin, will be glucagon, because as Bastiano said correctly, it's not a beta cell replacement, it's an islet cell. It's a mini pancreas inside of the capsule. We at Siglum, we are in the clinical phase with our first program in hemophilia, where no immunosuppression, patients have received spheres containing epithelial cells engineered to produce high levels of factor VIII for them. And we have a second program that will be starting this year on MPS1, where the same concept, continuous secretion of enzymes, in the case IDUA for MPS1. I think for diabetes is one level more in terms of complexity, which is the sensing and responding. If the cells are happy, if we are avoiding the perifibrotic overgrowth, this can last for many, many years. And of course, because you don't trigger the immune response, we can be redosed. So for me, really the goal is to have an insulin independence in the absence of immunosuppression, because that is what the patients need in the vast majority of patients.
Thank you so much, Rogerio. And it's true that the immunosuppression, I think we want to avoid, it also can impair the beta cell survival itself. So many reasons to avoid. And assuming that we will do that, and it sounds like we are on our way to cloak the cells against the immune attack and to make highly functional islets. I'm going to turn to you, Tom, because at that point, we really need to think about manufacturing, keeping the integrity of the cell, et cetera, and scaling the production up. So maybe you can start that question, and then we can bring it to the group to discuss that all-important question.
Right. So, there's a number of challenges that we're going to need to overcome specific to manufacturing. And I think it's sort of one of the last things people think about as they're doing their product development. But thankfully, these challenges are being worked on by a number of public-private partnerships, some in the US, like BioFat.
We're losing you a little bit, Tom.
USA that we're part of, Nimble, and CMAT. That's true not only of stem cell derived islets, but also a variety of cell types. And the challenges that we're facing fall into roughly 6 cross-cutting categories. More consistent raw materials, the things that Rogerio and Monsi talked about that were so great about the response of cells versus a device, is also what makes them so hard to manufacture. Their responses to everything they come in contact with. And so we need to be able to control that. We need to build enabling equipment that's designed for cells as medicine and not cells for making medicine. And integrate those technologies into closed modular and automated manufacturing systems, which we're starting to do now. And then non-destructive measurement and data management. So think about real-time sensors for things like, insulin secretion in response to glucose, or sterility sensors in manufacturing that'll allow you to do real-time release of products that might be stable for 48 hours and not really have a good shelf life initially. And then new technologies, we talk about cell survival and reimplantation. Part of that is getting the cells from the end of the manufacturing line to the patient. So we need new technologies that allow preservation and transport of a wide variety of living products, not just cells, but mini organoids, so stem cell derived islets, from the manufacturing line to the patient. And then really underpinning all of that is the need for data-driven process development. We've done so much guesswork. Luckily, islets, we understand, and Bastiano, I think, said that, the islets that are being produced at Vertex now are virtually indistinguishable from those from a cadaver. So we understand those pretty well, but all these products are going to need real process development work that's based on a lot of data, not hypothesis-driven activities.
It sounds like, in essence, you all need each other in the end. And everybody's developing a different aspect of this. But in the end, it's not just the cells. It's the technology and the delivery around it. So I guess the next question really for all the panelists came from our prior discussion around this, which is, what keeps you up at night and scares you when it comes to developing this technology as a therapeutic? And perhaps we can go around. We can start with you, Monsi, and then go with the rest of the panel.
Sure, and I think that Bastiano, Rogerio, Tom, we've all touched on this. So things that keep me up at night with what we're developing. We've learned a lot over the last few years being in clinic in patients. since 2014. And honestly, it's the same thing that keeps us up at night. So acute cell survival and the host response. So how, and again, we've touched base on this, but what are we doing as far as what are the challenges and how are we addressing them? So for the acute cell survival, we're thinking about specifically after implantation, how do we get the cells to survive? How do we allow hypoxia not to happen? Graft survival. The solution really is the things that we've talked about: delivery device, encapsulation. How do we optimize the encapsulation process? How do we get the membrane to be fully unrecognized by the body? And that really brings me to the host response as well, which is part of the challenges are the foreign body giant cell response, or even fibroblast reaction, as Rogerio had touched on. And that's really going to also be part of the solution is going to be the gene editing. And we're working with CRISPR to really figure out how to make these cells immune evasive as well. And again, the membrane that we have with our collaboration with the GOR. So the lessons we've learned are really going to inform the next steps. But again, they continue to keep us up at night because we want to optimize them as we're going in clinic.
Great. And Bastiano, what would you say about what keeps you up at night?
So I don't get scared easily. Otherwise, probably I will not do drug development. It's not for the faint of heart, especially in cell and gene. What keeps me awake at night is overall is a sense of urgency. I think that in the last few years, the field has accelerated, but I think there are still significant barriers when it comes to cell therapy in general. And now I'm speaking more of as a practitioner of the cell therapy field than just a type 1 diabetes. If you look at I started my career in cell therapy with now is Kymriah, so a CAR T. The concept that you can really treat a large number of patients requires a fundamental change in the way we think cell therapy in terms of manufacturing controls, what Tom was saying. So far, we have been really able to cure or to treat patients in predominantly pediatric rare diseases. The vision here is to really bring this type of curative intent to large number of patients like in type 1. So what keeps me awake at night is think about how I can scale up. Now I think we are fairly satisfied with the quality of our cells. As I said, our islets are virtually indistinguishable from cadaveric islets. Now we have to really step it up in terms of how many can we make. Can we make enough for all of those patients out there, as opposed to a few hundreds? I think that is the challenge in general for cell therapy, specifically for those indications that are really prevalent, as opposed to ultra rare.
Yeah. And Rogerio, what would you say? You touched on a lot of your concerns around scalability also, but.
Yeah, and I would not repeat this. Of course, when we move from the research into commercial, there is a big difference. We needed to repeat time over time. And again, at Siglon, we are really focusing our manufacturing process on really bringing cryopreservation, not only for drug substance, but also for drug product, automation of the encapsulation so that we can produce much more volume so that we can really have off-the-shelf product. Because once we have that, then we can really treat a much larger number of patients than we are treating today. I just wanted to make one comment on what Mansi said about the really oxygenation and location. I think that this is where we decided to go into the peritoneal space on the omentum, because there you have, one, the proximity with lots of very important organs, and number two, lymphatic and vascularization extremely rich over there, and that could allow really, one, a good nurture of the cells, but two, really a good exit for the proteins that we are producing to go where they have to go. But I would say that the really is, can we add the word that Tom said that I think is the most important one, control. Can we control the whole process? This is why we have a modular platform, so that we can really don't need it to invent small solutions for everything, but we follow in a modular way all the process. And of course, learning from the other programs, in our case, helps tremendously for the diabetes program.
That's really interesting. And Tom, what would you say, listening to your colleagues, about achieving that control? I know you mentioned you want everyone to be thinking about the manufacturing and scaling from the get-go. Is that happening, or what do we need to do next?
So the first step is thinking about all that early, right? So people leave it to, like I said earlier, people leave it to sort of the last minute. Thinking about, I guess the first thing I would say is data is essential, right? However you can get it. Everything is underpinned in cell therapy, as it is in drug, by a robust set of critical quality attributes. And those attributes, which are those attributes of the final product that correlate with final product safety and efficacy, right? So cells are putting out all kinds of factors constantly in response to their environment. So what we need to understand is which of those correlate with efficacy in the patient. The good news, as I said, for islets is that what makes them efficacious or functional in patient is really well understood. Mm-hmm. So once you have those critical quality attributes in place, then you can be sure you've got an anchor for scale up, for distributed manufacturing, for supply chain flexibility. It's not going to matter that you have to change your collagenase from one vendor to another, because you're going to have already figured out that that's important.
Right.
And that gets to then, it's not just about data, collection for critical quality attributes. You'll need to understand what aspects of your process influence final product quality, and then focus on measuring those steps. So that gets back to control during manufacturing, and then controlling those steps, then everything else is details if you can control those steps. And without this approach, which is going to require, we set up a core facility to study these very things, because it requires such a tremendous amount of data and instrumentation. But without this approach, we've come to live in this world where we say the process is the product. That can't be true, mm-hmm. right? The fact that we've landed on the exact process required to make islets that work, in the patient. Mm-hmm. I'm sure that across the board here, nobody's making islets the same way, and they're all efficacious, right? So, and that also forces us to live with sometimes impossibly tight manufacturing process specifications that don't give us any flexibility. So that's what keeps me up at night on all of your behalf.
We appreciate it, and I know the patients do too. It looks like we're going to wrap up with our panel here. I just want to say thank you to everyone. I know we didn't get to the big question around when will patients see this, but I know that we're there quite yet, and there's a lot of regulation and regulatory work to be done. But we're getting so closer. Thanks to you all. I want to say thank you again to Mansi Jaiman, Bastiano Sana, Rogerio Vivaldi, and Tom Ballenbach. And we appreciate you attending this year. And thank you once again.